Hello and welcome. Have you ever done an exam, felt like you knew your stuff, but then the grade didn't quite match up? Well, it's often about knowing what an examiner is really looking for. Today we're going to get inside their head. We're deconstructing a real AQA GCSE Biology question using the official mark scheme. It's all about learning the strategy to get every single mark you deserve. Let's get into it.
So this is the big question, right? You can have all the biology knowledge in the world, but to score those top marks, you have to serve it up in the exact way the mark scheme demands. It's a game of precision, and we are about to break down all the rules for you.
Our focus today is question 3 from an AQA GCSE Biology paper. And honestly, it's a perfect example because it tests a little bit of everything. You got practical skills, experimental design, and some core biology knowledge all rolled into one.
Okay, so here's the whole question up on the screen. It starts with a big 6-marker on chemical tests for carbohydrates, and then it gets into a student's investigation about why bread tastes sweet. We'll be looking at variables, validity, the whole shebang. Feel free to hit pause right now if you want to read it all through before we start.
Let's start with the beast, the 6-mark extended response question. I know, these can look a bit scary, but they are so achievable if you just have a clear structure. We're going to build the perfect answer together using the exact phrases that get you the points.
So the question is asking for 3 key things: the test for one type of carbohydrate, the test for the second type, and a risk assessment. If you miss any one of those, you're putting a cap on your marks before you even start. And look at that command word, describe. That means you have to say how to do the tests, not just name them.
First up, starch. Now the mark scheme is looking for some very specific things here. You get a mark for naming the right chemical. Iodine solution. Simple enough. Then you get marks for the color change, and this is crucial. You have to state the starting color, yellow, orange, or brown, and the positive result, which is blue-black. You need both to lock in the marks.
Okay, next up is the test for sugar. First, name the reagent, Benedict's. Now, a key detail here, and this is a classic mark that so many people miss, is that you must mention that you heat the sample. You have to. Then you describe the color change, from blue to this whole rainbow of positive results, anything from green all the way to brick red.
Seeing it all laid out like this in a table? Super helpful for getting it stuck in your brain. This is just one of those things you have to memorize. And notice, for the Benedict's test, you've got a whole range of positive colors, and naming any one of them would get you the mark.
And finally, the risk assessment. The question specifically asks for it, so you can't ignore it. Don't throw away these easy marks. The mark scheme wants you to identify a risk and say how you'd manage it. For example, risk of burns from heating? Precaution, use a water bath. Risk of chemicals spitting in your eye? Precaution, wear eye protection. Simple, but absolutely vital for hitting those top marks.
And here it is. This is the secret sauce. This is a direct quote from the examiner's instructions. To get into that top band of 5 or 6 marks, what they call level 3, your answer has to have all 3 of those parts we just talked about: the starch test, the sugar test, and the risk assessment. It's literally a checklist.
All right, let's switch gears and move to the next part of the question. We're now looking at the short answer, single mark questions. Now, they might only be worth 1 mark each, but trust me, they add up so quickly. This is often what separates the grades. First up, a question on experimental design, asking about the dependent variable. So the student was timing how long it took for bread to taste sweet. The thing being measured is the dependent variable. But the key here is precision. Just writing time isn't going to cut it. The mark scheme is super clear. It says to ignore time unqualified. The only way to get the mark is to say time taken for the bread to taste sweet. See how specific that is?
Okay, next up, control variables. In other words, what should the student have kept the same to make it a fair test?
The good news is the mark scheme gives you a whole menu of options here. You could have mentioned the size of the bread, its surface area, where it was placed on the tongue, or even making sure to clean the mouth between tests. You only needed to write down one of these to bag the mark.
I love this kind of question. It's asking you to think like a scientist and critique the experiment. Why might these results be a bit dodgy, or, to use the scientific term, not valid? And notice, it gives you a big hint. Don't talk about the control variables again.
And again, you had a few ways to get this right. You could point out that the experiment wasn't repeated to make sure the results were reliable. Or, and this is a brilliant point, you could say that taste is subjective. What I think is sweet might be different for you. Or you could point out that the amount of saliva someone produces can change. Any of those would have been a perfect answer.
Okay, time for the final piece of this puzzle. This is where we explain the actual biology behind what the student was observing. It's a 3-marker, so it needs a clear, logical story.
So the command word here is explain. That's your big cue.
You need to give the why. Why does a piece of starchy bread start to taste sweet after you've been chewing on it for a bit? Let's break it down.
Now, this is super interesting, and it's a really important peek behind the curtain of how these exams are marked. The examiners know there's often more than one right way to explain something. For this question, they actually laid out two totally different, totally valid routes to get all 3 marks.
So, Route 1 is probably the classic answer you'd think of first. Point 1, you state that bread contains starch. Point 2, you say that this starch gets broken down into sugar. And for the killer third point, you have to name the enzyme responsible, amylase, which is in your saliva. 1, 2, 3, a perfect chain of logic.
But check this out. Here's the alternative. You could also argue that bread already has some sugar in it. So, mark 1, bread contains sugar. Mark 2, this sugar dissolves in your saliva. And mark 3, that sugary solution then reaches the taste receptors on your tongue. 2 different explanations, both completely correct, both worth full marks. Pretty cool, huh?
Phew! Okay, we just took apart that entire question, piece by piece. So what's the big picture here? Let's zoom out and think about the strategy. What's the mental checklist you can use for any exam question you face?
First, you have to read those command words like describe or explain and know what they mean. Second, you've got to know your chemical tests inside and out. They're such a common topic. Third, if they ask for a risk assessment, you give them a risk assessment. Fourth, be so precise with your scientific words. And finally, really get your head around the ideas of experimental design, like what variables are and what validity means.
Look, getting good at this isn't some kind of trick to cheat this system. It's about learning to communicate your knowledge with total clarity. It's about speaking the examiner's language. So the real question for you is, now that you know the rules of the game, how are you going to use this new level of precision on the very next practice question you try?